Okay, so just as we get started, a little bit of housework just to keep the background noise down. We are going to keep the audio from all the attendees muted during the presentation, but we do encourage you to ask us questions as we go along, and you'll find a Q&A button on the Zoom meeting toolbar that will allow you to post those questions as we go. And we'll also have a question and answer session at the end of the webinar to address all of those questions. So Sam's given you a little bit of an introduction to me, but I'll just briefly introduce myself a little bit more. I do live in New Zealand, in Auckland, where Kaha Science is primarily based. And I have a PhD in physiology and electrical and electronic engineering from the University of Auckland. I've officially worked in telemetry since I joined Telemetry Research in 2009, but I was involved in the laboratory where the telemetry system was first developed and then commercialized in 2004. And now I'm the Chief Scientific Officer at Kaha Sciences, where my role is to provide scientific support to our staff, distributors, and most importantly, to the users of our telemetry systems. As well as my role at Kaha, I still hold a 0.5 position as a Senior Research Fellow at the University of Auckland, where I'm a member of the Department of Physiology and the Bioengineering Institute. Now, with a background of physiology and engineering, my university research involves the collection and analysis of physiological data from animal models in both control and disease states, much of it using telemetry. My current research focuses on the interactions between arterial pressure and intracranial pressure in normal and hydrocephalus or raised intracranial pressure animal models. And I've developed techniques to chronically record intracranial pressure in both rats and sheep, which allows us to take advantage of the benefits of working with both small and large animals in our studies. This recent focus on intracranial pressure has grown from a project to develop a fully implantable clinical device to allow chronic measurement of intracranial pressure in patients with hydrocephalus. And we are now also applying similar technologies to the development of an implantable light source for optogenetics therapies and are exploring its application in both small and large animal models. So we can now record a range of signals from both rats and mice using telemetry. And here are some examples of the signals recorded using the Kaha Sciences telemetry systems. The beauty of telemetry is that these can all be done in conscious, freely moving animals living in their home cages. And this means we can design experiments to address important scientific questions without the data being confounded by anesthesia or handling stress. As a quick introduction to our telemetry systems, we have telemeters available for both rats and mice. The telemeters are paired with a receiver and wireless power platform, which is called a smart pad for the rats or a T-base for the mice. The smart pad or T-base is placed under the animal's cage and receives the digital signals from the telemeter, recreates the analog signal, and outputs it to a data acquisition system. Because our systems use digital transmission, you can record from up to 40 telemeters at a time without any crosstalk, interference, or without the need to shield your cages. Recreating those analog signals means that the smart pads or T-bases can be connected to any data acquisition system that takes analog inputs. Now in this webinar, I primarily use AD Instruments LabChart as an example of data acquisition software because that's what I am most familiar with, but similar approaches can be applied to many other data acquisition systems. One of the key benefits of the Kaha system is that we use wireless power for both our rat and mouse telemetry systems. The SmartPad and T-base generate a magnetic wireless power field which passes through non-metallic surfaces such as the cage or the animal body. A pickup coil in the telemeters uses that magnetic field to produce a current to power the telemeter and, in the case of the rat, charge the battery. This means that experiments are no longer restricted by battery life and that telemeters don't need to be sent back for refurbishment between uses. If you're interested in learning more about the technology behind our telemetry systems, I encourage you to view the recording of the webinar titled Telemetry 101, which is available on the Knowledge Centre section of the Kaha Sciences website. So here's an example of pressure data recorded from a rat via telemetry. The red line at the top is arterial pressure recorded from the abdominal aorta, while the blue line is left ventricular pressure. The green line at the bottom is the PDT, or the rate of change of pressure, which is calculated from the LVP signal. Our telemeters sample signals at 2 kHz, which you can see gives a lovely high fidelity set of signals. That means that we will usually set the data acquisition system to sample at around 2 kHz. That is 2,000 samples per second. So you can imagine that being able to record 24 hours a day, 7 days a week, for potentially months, you have the potential to record a lot of data. Now this is great, and it gives the opportunity to design some amazing experiments, but it is worth considering the impact of recording all that data. And this is what I'm going to address in today's webinar. My colleague Dr. Phil Griffiths covered a number of things to consider regarding experimental design of chronic telemetry studies in the first webinar of this series. If you missed it, I encourage you to view the recording. It's available on the Knowledge Centre section of the Kaha Sciences website, or our Syndica also have a link to the webinar on demand on their website. Okay, so in today's webinar, I'm going to cover 3 main topics regarding handling of the large amount of data that you may record during telemetry experiments. First, I'm going to look at the possibility of automatic scheduling of those recordings. I'm then going to discuss some options for automatically saving the data files and managing your data before I move on to some suggestions of how you might like to automate some of those repetitive data analysis tasks that we all have to do. As I've said, I'm using the 80 Instruments LabChart data acquisition software as an example because that's what I have used most. But hopefully it gives you an idea of the approach you can take, and that can be applied to other systems as well. Okay, so let's get started. One option to cope with the large amount of data collected from a telemetry system is to limit what you record or save in the first place. I guess you could call it a minimalist approach. If you don't need it, don't record it. But how much data should you record? How do you know what you need to record? But the very first thing to consider is the hypothesis of your study. What questions are you trying to answer, and how will you present your data to address those questions? Are you expecting to plot daily averages over time? Or are you interested in how the signal varies across the course of a day? What statistical tests will you apply to your data, and what data do you need to be able to test your hypothesis? And of course, we also need to keep in mind the signals that we are recording. What sampling rates should you use, and how do those sampling rates vary over time? So by automatically scheduling the data recording, you can spread the data throughout the day and the night, giving a better representation of the true mean. The example graph here shows the effect of 2 different sampling protocols on arterial pressure. For simplicity, the data shown are by the little black dots are 30-minute averages. The solid line shows a cosine fitted to the dots and shows the circadian variation in the blood pressure across the 24-hour period. These blue triangles represent one sampling protocol in which 6 hours of data, or in this case 12 points, are recorded during a single period between midnight and 6 a.m. Whereas the red circles represent a protocol in which one sample is taken every 2 hours, spreading those 12 sampling points across the whole 24-hour period. You can see the difference in the ability of recording for 30 minutes every 2 hours to represent the circadian variation compared to doing all of that recording in one single 6-hour period. Given the impact of spreading the data across the 24-hour period, automation of the scheduled data recording becomes really important. This allows the collection of data to continue even without you being present in the lab, and the data can be set to record at the same time every day, making analysis easier. The biggest advantage to scheduling data recording is, of course, the reduced volume of data that you must then store and analyze. Now, we thought quite a bit about this question a few years ago when we were recording data from rabbits via telemetry. We noticed that all of the data we recorded over the 4 to 5 weeks of our experiment generally boiled down to presenting daily averages once we analysed it. We realised that perhaps we didn't need to record data for 24 hours a day, every day, and we set out to test that effect. We applied various recording protocols to pre-recorded data to see what error we got when looking at the 24-hour mean of different variables. I'm going to show you the effect on blood pressure data here, but more information is available in our article published in the American Journal of Physiology. So to quantify the effect of different sampling protocols, this graph shows the percentage error of calculating the 24-hour mean of arterial pressure using those different sampling protocols. The total recording time is plotted along the x-axis, and on the y-axis we have the absolute error in calculating that 24-hour mean of arterial pressure. The sampling protocols are then separated into the number of recording periods each day. So along the top here, you have the effect of recording, for example, 2 hours of data in one single data period or one chunk of data, compared to spreading that over 12 recording periods, or 12, starting and stopping recording 12 times. You can see that in general, the more recording periods per day, as you come down, gives you a lower error. And if you look closely, here you can see that once you get to about 2 hours of data recorded, there's actually minimal benefit of adding any more data to that recording protocol. What has the biggest impact is spreading that data across that 24-hour period. So you might, for example, choose to record 5 minutes of data every hour, giving you 2 hours of data across a 24-hour period, or rather, which in this set of data would have given the 24-hour mean of blood pressure with only a 1% error. So data scheduling in LabChart is done using what's called the scheduler extension. If you are a LabChart user and you can't see the scheduler listed under the setup menu, then you will need to install it using the feature manager. Using scheduler, you can choose how much data to record and how often. You can also force the data to record at the same time every day. For example, here I have set it to record for 5 minutes every hour on the hour. You can even set it up to record data to different LabChart files in turn so that your individual experiments can go in different files. I always make sure to check the autosave box at the top so that my data is recorded after each recording period. And in this case, if you choose the everyday option, then once you start it, the scheduler will continue to run until you stop it. Now, another recording system that I am familiar with is LabScribe from iWorks. And this allows you to choose the acquisition mode in the preferences menu. The parameters are similar to what I've just shown you, but the terminology is slightly different. And they also have a macro function similar to one I'm about to show you in LabChart, which allows for a lot of flexibility in the configuration. And another popular data acquisition software is CED's Spike2. Now, Spike2 uses a powerful scripting language to configure and customize their software. Example scripts are available for download on their website. This particular script, or family of scripts and sampling configurations, is very useful and allows you to perform long-term recordings of blood pressure, heart rate, and sympathetic activity to analyze baroreceptor reflexes and heart rate variability. The scripts are suitable for use with a range of laboratory animals and can record from up to 8 animals simultaneously when used together with a telemetry system. The script derives systolic, diastolic, and mean blood pressure, heart rate, and respiratory rate from each blood pressure trace. The data can be recorded from up to 8 animals and then analyzed in either online or offline. Results are recorded continuously or recorded according to a user-defined schedule. For example, you can set it to do 10 minutes per hour. Data can be auto-saved to a new file at regular intervals, for example, every 24 hours, in order to achieve virtually continuous recording over days or weeks. So when thinking about how much data you need to record, you also need to keep in mind the stability of your signal. As shown earlier, scheduling of arterial pressure signals will likely give you a good representation of the 24-hour mean, but it is generally a very stable signal, and we are often only looking for slow changes over time. In one of our other studies, we were recording left ventricular pressure in rats, and we found that the signal was more prone to artifacts from the catheter position changing with animal movement. That meant that we could not always get good data from every recording period. And it was important for us to account for this by recording more data than we might have otherwise to ensure we got a good spread of data across the day. For example, we wouldn't have wanted to lose all of our nighttime data when the rats were active, as this might have skewed our results. We also made sure to visualize the data as we analyzed it to make sure that the waveform shape was acceptable and therefore the data calculated was valid. Now, of course, if you're trying to capture things other than the 24-hour mean, scheduling your data recording might not be appropriate at all. Detection of seizures or arrhythmias, for example, would be better done with continuous recording to avoid missing those events. You may also be planning other types of data analysis, which may place other constraints on your recording protocol. For example, if you wanted to perform spectral analysis to look at low frequency oscillations, you will need to make sure to record enough data in each sampling period to get a long enough segment of stable data to perform that analysis on. Now just remember, there's no right answer that can be applied to all circumstances. At the end of the day, it's your data and your experiment. So data storage has become a lot more portable, cheaper, and accessible over the last few years. So the option of recording data for 24 hours a day, 7 days a week is becoming more attractive and much more practical. There are still some challenges involved with collecting that large amount of data, but they have changed from being where to actually store it to how to keep track of which files are which through the naming of files, and how to make sure that the data are saved in the appropriate sized files. Automatic file saving is an easy way to achieve consistency with your files. Files can be set up to automatically include the date and time the file recording started by using it in the file name. It is also convenient to make sure that the files don't get too big to be able to analyze them. I used to manually save my data to a new file each day, but sometimes that would get missed, for example, on the weekend. And the files would get very large. And in my experience on my computer, once lab chart files get over, say, 1 gigabyte, it can be very difficult to work with them to perform any analysis. Keeping your file sizes and time periods consistent can also make your data analysis easier. So I'm again going to use LabChart as an example here, but similar procedures can be applied using scripts in Spike2 or macros in iWorks LabScribe, for example. And I'm sure that other data acquisition systems also have ways to automatically save files. I'm hoping that by leading you through the way I do it in LabChart, it will give you an idea of how to approach it in whichever system you are using. So in LabChart, we can use macros to automate a vast array of tasks, including file saving. I have written a macro to save my data to a new file every 12 hours. The instructions for this macro can be downloaded from the Kaha website. Briefly, you set up the LabChart file as you would normally, including the units conversion, sampling rates, etc. I also suggest that you set up the columns for the data pad for the analysis you think you might want to perform. For example, you might like to set up a column to calculate the heart rate from an ECG or arterial pressure signal. In general, I avoid setting up channels using the more complicated data analysis extensions in LabChart, such as the blood pressure module at this point. I have found that it slows down any scrolling through the data as it recalculates for every view. Plus, I'm often recording from several animals at once in the same file, so I prefer to make copies of the files to apply that analysis to the individual signals at a later date. Once your LabChart file is set up, you can then append the settings file containing your macro, and that will add the macro to your LabChart file. And as I said, there are examples of these macro files and instructions available on the Kaha Sciences website, and you can download those and customize them to your application. So the way I set up the macro, you'll have to open the macro and make a couple of simple changes before you run it. There are details in the PDF instructions on how to do that opening and editing of the macro. Basically, you need to edit the path highlighted here to tell the macro where your data should be saved and what to call the files. You can then also change the length of the recording period if you wish. LabChart uses seconds as its base time unit, so 43,200 seconds is the equivalent of 12 hours. So the macro starts by, firstly, saving the file using the file name provided with the date and time appended. It then starts sampling of the data and counts down the 12 hours or however long you have chosen to record for. At the end of the recording period, the data file is saved. The data are selected and then cleared, and then the macro repeats, starting with saving to a new file name. So you can see in the title bar of your file how much time is left to run in the current recording periods. You can still continue to add comments and to view your data as usual, and the macro will continue to run until you press the stop button in the playing macro window. What you end up with is a file every 12 hours named with the date and time that file was started. In this example, I have recorded 3 channels of data from each of 3 mice, which gives me 9 channels. 12 hours of recording of 9 channels at 1 kilohertz gives around 300 megabytes, which is well within comfortable limits for later viewing and analysis. So if you're recording video in LabChart, then you need to run a slightly different version of this macro. And that's because LabChart asks for confirmation before clearing any data containing video. I've created a version of the macro that calls what we call a VBS script to automatically acknowledge this request, and that's also available on the Kaha website. As I've already mentioned, keep file sizes under consideration when deciding how long to record for in each file. Now, one disadvantage of this method is that you can only ever see a maximum of the last 12 hours of data when you scroll through the data while it's recording. This means that to keep an eye on the trends in your data, you may need to regularly view your data offline and do some basic analysis as you go. I also encourage you to think about where you are storing your data files. A local PC drive may be the best option for day-to-day recording rather than across a network, but you also need to make sure that you move or copy those files to a server or cloud-based storage regularly.
Thank you.
Okay.
Well, sorry. I think we'll launch an audience poll at this moment and give Sarah Jane a second to catch her breath. So the second audience poll, which parameters from a telemetry implant would be most interest to you? Of the options that we have available, we have ECG, EEG, EMG, blood pressure, sympathetic nerve activity, tissue oxygen, temperature, activity, or optogenetic stimulation. Which of these parameters would you be most interested? While everyone is answering the poll question, I would like to encourage all those who are attending to submit any questions or queries they have that we will address at the end of this presentation. Perhaps some information that Sarah Jane has mentioned in her topic has piqued your interest. You may want to know more. Please submit that at the very bottom. It seems that the majority of people are interested in blood pressure and activity, which are also some of the most common parameters mentioned. So I'm going to end this poll, and I'm going to bring in back Sarah Jane to finish the rest of her presentation.
Thanks very much, Sam. Okay, so far I've showed you how to use macros to automate your file saving. But macros can also be used to automate the analysis of your data. And they're particularly useful when you need to apply exactly the same process to a large amount of data. Now again, I'm going to use LabChart as an example, but similar approaches can be applied to other data acquisition systems. One example of a repetitive task that I seem to apply to almost all of my data is the calculation of hourly averages. So I've written a macro to help me do this. I put all the files that I want to analyze in a single directory and then run the macro to process the files. This time, rather than appending the macro to my data file, I have a LabChart file that I open and run the macro from within that. And again, there's instructions in the LabChart files are available for download from the Kaha website. Now, I'm not going to go into details of the code, but the macro steps are that it first asks for the location of the folder that your data is stored in, and then it opens each of those files in turn. For each file it opens, it adds a date and time columns to the datapad and sets the time mode to time of day. Now, this is mainly because I forgot to set those up when I first created the first data file, but I've left it in there because it also gives you an idea of how you can use macros to set up new datapad columns if you need to. The macro then runs the multiple add to datapad function to select 1 hour of data every hour and adds the data to the datapad. Now in LabChart, the columns you have set up in the datapad will determine what data you get out. By default, they are set to do an average of each channel, which is really helpful, but you can add others. For example, you might like to use average cyclic rate to calculate heart rate from your blood pressure or ECG signal. The macro then saves the datapad as a text file, and it closes that lab chart file. Lab chart, when you close a data file, asks you whether you want to save that data file. So I've got around this by again using the macro to call a VBS script to say no. The macro then moves on to the next file until all the files in that folder are processed. The output of this macro is a text file here for every lab chart file that was analysed. Now, as you can see, I haven't bothered to force the names of the text files to anything sensible, and that's because the next thing I do is to run what we call a batch file to combine the individual text files into one larger text file. I can then open that combined text file in something like Excel and sort the data by the date and time columns, and that puts the data in the correct order. It is then all ready for me to plot and do further analysis on. Now, if hourly averages isn't what you want to calculate, then you can create your own macros to automate just about any task in LabChart. Even if you don't have any programming experience, you can use the record macro function. You simply press record, do the steps you are wanting to automate, and then press stop. The macro commands for each of the steps you have just performed will appear in the macro text. I recommend always starting with a new macro window so that you know which commands are associated with each recording. You can then edit the commands in the macro text and combine them with others to create your own macro. A few words of caution though. Don't over-automate your analysis. It may seem easier to calculate the 12-hour average directly from the data acquisition system and just plot that data. But I like to plot the hourly averages in Excel and then do further averaging from there. That way, I can see the effect of the circadian rhythm, and I'm also more likely to notice if one of those data points is an outlier and may affect the mean. Secondly, don't leave all your analysis until the very end of your protocol. For very long-term experiments, for example, those lasting for months with slow-moving trends, I like to analyze the data in 1-week batches and plot it as I go. That way I can see the trends as they emerge. However, if I'm expecting things to change more quickly, I will of course plot and review the data more often. Most of my experiments involve recording a baseline data period and then performing an intervention to induce a change. And I will always plot that baseline data before moving to the next stage of my experiment. There's nothing more frustrating than analyzing your data at the end and realizing that your baseline wasn't quite stable and that you should have waited an extra day or two. And of course, it's still really important to check both your signals and your animals every day. So that's the end of our webinar for today. Hopefully this presentation has got you thinking about ways to improve how you work with your telemetry data. My key tips for making life easier are to always consider the questions you are trying to answer and how you will present your data. Automate your file saving to stop the files from getting too big, and set up your data files properly right from the start with consideration of how you will analyze your data. Use macros or scripts to automate those repetitive data analysis tasks, and always back up your data. So just a quick reminder that you can download the instructions and example files from the LabChart of the lab chart macros I've introduced today from the Kaha Sciences website. I also encourage you to check out our Knowledge Centre section of our website, and there we have recordings of previous webinars, surgical videos, and lots of other useful information. Registration for the Knowledge Centre is free, but approval is a manual process. So depending on the time of day, you may experience a short delay before your registration is approved. Now, all of the data acquisition systems that I've mentioned today are supported by great teams of people who are a lot more familiar with their software than I am. They have all been really helpful when approached to troubleshoot and provide advice on how to work with their systems to collect telemetry data. And I encourage you to reach out to them to discuss what you want to do and how it might be done. And as always, if you have any questions, please contact us on support@kahasciences.com. Okay, so I guess we can see if we've got any questions, Sam.
Yes, thank you, Sarah Jane. And we'll take this opportunity to ask any questions that have been asked through your presentation. And I encourage, again, others to ask questions in the Q&A dialog box below. During your presentation, Sarah Jane, some attendees did ask a few questions, and I'll start off with the first one. One of the attendees asks, 1 kilohertz is quite a fast sampling rate. Couldn't you just lower the sampling rate and make the file smaller that way?
Yeah, that's a really good question. And I've actually borrowed a slide from Phil Griffith's first webinar in the series to help answer that. And what you can see here is an ECG signal from a rodent plotted at 3 different sampling frequencies. And you can see the top one is 2000 data points per second, 2 kilohertz. And if you downsample that to say 400 data points or 200 data points per second, you can see the effect on the signal. So a lot of our signals, particularly because we're working in rodents which have a very high heart rate and very fast moving signals, are best captured by the high sampling rate. So if you're using something like, for example, our tissue oxygen telemeter to record tissue oxygen, that's a much slower sampling rate. So you would be able to drop your sampling rate in that case. But for something like blood pressure, ECG, or particularly left ventricular pressure, then a higher sampling rate will give you a much better representation of that waveform. And you can see that even if you are interested in, for example, just the RR interval to calculate heart rate, if you downsampled your ECG signal as low as 200 data points, you still see the blip, but the timing of that R wave, you're missing that peak. And so your timing of those RR intervals is not going to be accurate. So I do think the beauty of telemetry is the high frequency sampling we can do, but you do then have to think about those other ways of managing that data.
Agreed. Another attendee mentioned, I guess, in their labs that they notice a peak in their blood pressure and heart rate data when they disturb their rats for feeding and cage changing. What should they do about this when they're analyzing their data?
Yeah, that's another good question. Now, a lot of people have this issue, and I guess that's again where telemetry comes into its own, because if you can see the effect that handling of animals and disturbing animals has on their variables, particularly those of us that are doing things like cardiovascular studies with heart rate. And what a lot of people will do is make sure that all of those animal husbandry tasks are done between, say, 8 a.m. and 10 a.m. in the morning. And they will, rather than doing a 24-hour mean, they might choose to do a 22-hour mean. And as long as you get it consistent, you can actually often see that peak occur at the same time every day. And so if you're consistent, you may want to just leave it there and include it in your data. And as long as you're aware that it happens in every animal, or as I said, you might want to just change it to being to exclude that 2-hour period every day and look at, say, it's 22-hour mean.
Okay, I see. Another attendee asked a question about the macros that you presented. They say that the macros seem very complicated. Do I need to be able to understand code to be able to use them?
Yeah, that's a good question. Um, the short answer is no. I mean, I have an engineering background, and I'm sure it helps with particularly the confidence to play with those macro files and edit them. But if you're not familiar, they are in, let's say, almost plain English. And I think you can pick up enough from each line of the macro code to know what that line is contributing to the macro. And you might not necessarily know what the different parameters that finish that line are, but so long as you know which line you're referring to, and you can see when you have a play, when you change things and you're recording those macros, you'll soon see the different commands come up and what they do. And if you change a value, for example, you'll see that change in that code, and it's easy enough to then move them around. And I encourage people, just have a play, save your data, save a copy of your data. You can't do anything wrong,
Hmm.
and have a play and see what happens.
Yeah, see, I think I agree with you. The working on a copy, it can't go anything wrong with that.
That's right.
Another attendee asked, how soon after implementing the telemeter can I start recording data?
Mm-hmm. So to implant the telemeter, obviously the animal has to undergo surgery, which involves an anesthesia and all of those sorts of things, and that will disrupt their physiological systems. And so often what you'll see is that blood pressure and heart rate in particular are higher for a few days after the surgery. So what we do is we still record our data, but we monitor what those blood pressure and heart rate are doing. And in particular, what you start to see 3 or 4 days after surgery is the circadian rhythm come back in heart rate and perhaps blood pressure. And that's a really good indication that your animal is recovering well from surgery. It's starting to move around at night more, and you start to see that increase in heart rate. And so what I would recommend is that you give the animals a good week before you start recording your baseline data. But again, I stress the importance of plotting that baseline data period to make sure that everything has stabilized before you move on with your experiment.
Okay, very interesting. Sarah Jane, I know you have the option to open up the Q&A dialog box. A
Mm-hmm.
few questions I've entered there. One of the questions seems very interesting. The attendee asks, when dealing with pattern recognition software for ECG analysis, what is the best approach to teach the system what a basic arrhythmia such as PVC is, especially when you can have a great variation of classic PCV waveforms? Do you have any experience or can that talk?
Yeah, that's a little bit outside my
Mm,
experience, but I can say that I do know that LabChart, for example, has quite strong ECG analysis algorithms, and it can allow you to do some averaging of those ECG signals, and it will automatically detect various components of the ECG waveform and allow you to calculate the timing of those. And so I think that will pick up a lot of those arrhythmias, and you can look at, they can do scatter plots which show you the each beat and where those lie in terms of the different timing of those waveform components. And that's a really good way of detecting whether you've got arrhythmias. And they also all have most of the data acquisition software packages that I've mentioned have pattern recognition and like template recognition. So you can go along and detect an event based on the shape of a certain waveform. But again, I encourage you to reach out to the software developers, because they have a really good handle, and they are all really interested in helping people to work out how they can do various new data acquisition and analysis processes with their data. So they're a really good source of information for something more specific.
it's good advice, especially there's another question that just came in similar about that, but for AFib location in 24-hour recordings, any
Yeah, no, again, I would anticipate, I haven't done it, but I would anticipate using something like the event detection. If you've got a template that shows what you can, the specific pattern that you're looking for. There are also more complicated engineering type analysis with cross correlations and things like that that you could do offline as well in terms of pattern recognition. But I would have a look at some of those event detection algorithms and processes within your data acquisition software.
Right, right, right. Another attendee asks, did you measure LV pressure in freely moving animals, such as in rats? And if you did, was the catheter stabilized in the same position in the heart for the whole measuring period, or what instruments were used?
Yeah, so the video I showed you of arterial pressure, left ventricular pressure in a rat, that was done via one of our telemetry instruments, the 54 pp, which is a dual pressure telemeter. And the advantage of those telemeters is that they have a Miller solid state pressure sensor, the microtip pressure sensor, right on the tip of the catheter, so you can put it exactly where you want to record pressure. And that is how we've been able to get such great recordings of left ventricular pressure. And so the way we did that was to place it, the telemeter gets placed in the abdomen of the rat, and we go up through the diaphragm to the apex of the heart and secure the pressure catheter through the apex into the left ventricle. And there's some, if you have a look at the Knowledge Centre section of our website, there's a whole lot of surgical videos that walk you through that process. And we've also got a journal article that was published. If you search for my name and left ventricular pressure, you'll find an article based on the that method, and it will walk you through all the different waveforms and things like that. It's a mostly stable signal, but as I said, occasionally, because the animals move around, you do get some periods of artifact of the signal, but with a bit of practice, you can minimize those and get a really nice position. We've recorded out to 12 weeks in rats for left ventricular pressure, so it's a really good way of looking at cardiac function.
That's a good, yeah, good way. There is a user, sorry, an attendee that asks about power spectral analysis. Do you have any experience with that?
Yeah, I do. So I'm most familiar with LabChart, but I know that both LabScribe and Spike 2 also have the ability to perform it. And so you can select a period of data and perform spectral analysis. It's very easy. There's a button that does that for you effectively. You can select your nice stable period, and it will perform that spectral analysis. And it's a really great way of looking at the different frequencies that are occurring in your data and how those might change over time. So all of the data acquisition systems that I have mentioned today will do spectral analysis.
Mm, okay, cool. Good to know. Another attendee asks, in your experience, especially with the Kaha telemetry telemeters, which parameters are the most data heavy? Um, I would assume perhaps blood pressure recordings, or are they all kind of on par?
Um, I think probably something like left ventricular pressure in my experience, because you can calculate things like the dPdt, the rate of change. You can look at
Mhm.
end diastolic pressures. You can look at the peak pressures. So that's the one that I have kind of had the most experience with, and that's where the blood pressure module from LabChart works really well, because it automatically again detects all of those parts of the waveform and outputs that information in a table for you. The other one, I guess, is something like EEG, where the mean of the signal doesn't necessarily mean much, but you're then doing something like spectral analysis to look at the different frequencies of that signal and how they change over time. So that requires a little bit more, I guess, not necessarily thought, but a little bit more data processing and analysis to be able to pull that data out, rather than just running through and doing a hourly mean or a 24-hour mean.
Okay, that makes sense. And thank you for that answer. We're reaching that 60-minute mark, and I still encourage those to ask questions as we are going to wrap it up. And I would like to thank Sarah Jane, of course, for the wonderful presentation and for her thoughtful answers. As I mentioned earlier, we will be sure to answer anyone's outstanding questions privately in person, and we will also publish all the questions that were asked in a written transcript from today's Q&A session. Again, I'd like to thank Sarah Jane for her insightful talk. And I'm very hopeful that everyone walked away satisfied that we met your learning objectives today by reviewing how scheduling of data acquisition can be used to reduce the amount of data collected while still ensuring the physiological changes are accurately captured, how macros can be used to automate data acquisition and file saving to keep file sizes manageable, and how automatic processing of data files using macros can perform basic analysis quickly and easily. Please feel free to reach out to us directly or to Sarah Jane with any questions about what was presented today. As a reminder, our next session, titled Therapeutic Relevance of Elevated Blood Pressure After Ischemic Stroke in the Hypertensive Rats, is scheduled for 1 week today on Wednesday, November 4th, 2020, where Dr. Pratik Thakar from the University of Auckland will discuss, amongst other things, measurement of tissue oxygen, intracranial pressure, and blood pressure by telemetry in a rat model of stroke, how pre and post-stroke treatment of hypertension affect post-stroke outcomes, and the benefits and challenges of performing telemetry measurements and considerations for experimental design. I encourage everyone to visit syntica.com/webinars to learn more and to register for this upcoming webinar. You can get that information on the slide in front of you. Thanks again for everyone for taking time out of your day to attend our session. We look forward to seeing you at one of our upcoming events. Have a great day.